Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 2 for June 2022. This is the part 2 video of this paper, and I'll put the link to part 1 below the description box. Let's begin with question 5. It says, the Hawaiian Islands have 18 species of honeycreeper birds. They say the Hawaii Amakihi honeycreeper and the Hawaii Palila honeycreeper are two species found on these islands. The table gives information about these two species of honeycreeper. So they've given us the photographs for one. This is the Amakihi honeycreeper and this one is the Palila honeycreeper. They've told us about the habitats. This one here lives in lowland and mountainous forests and shrubland, while this one here lives in the mountainous areas. We can see that this one here has a sharp beak, while that has a very short beak. And they've told us the habitats. This one lives in more habitats than that one. And the population here, it's about less than 10,000, while this is less than 1,000. So here, this one has a higher population than the other one. The food source, this one here eats nectar, tree sap, spiders, and insects, while this one eats seeds and berries. So this one has a wider variety of food in comparison to that. So moving on, they say, the Hawaii Palila honeycreeper is found only in the mountainous forest in one island. Which of the following is the term used to describe this distribution? Since it's found only in that specific area, it should be endemic to that area. So the answer here should be a B. Moving on, they say, in 2003, it was estimated that there were 7,100 Hawaii Palila honeycreeper birds on this island, and in 2020, it was estimated that there were 950 of these birds. They ask you to calculate the percentage change in the estimated population of these birds, and they want you to give your answer to two significant figures. So here we have to know that since it's a percentage change, you can see the difference is that minus that gives you what change has occurred. So that is the change divided by the original population times 100. Here we get 86.619. When you round that off to two significant figures as required, we get 87%. So the answer here should be 87%. Let's move on. Here they say, it is suggested that both of these Hawaiian honeycreeper species evolved from the same common ancestor. Then they ask you to explain how these two species of honeycreeper could have evolved from the same common ancestor. They want you to use the information in the table and your own knowledge to support your answer. So here I said, both had a common ancestor who had a less powerful beak than Palilas and a shorter beak than Amakihis. If you look at the images, you can see one of them has a sharp. Let me take you back slowly. This one here has a very sharp beak, and this one here is kind of strong and short. So we can see it means their ancestor must not have had such a very long beak, and this one here, the ancestor wouldn't have had a very strong beak. So it was in between the two. A little bit long, but not too long, and it could have had some strength, but not too strong. So let me take you back. So I said colonization of the Hawaii Islands was a change in the environment. So the ancestor moved to this area, and there was a change in the environment and the conditions. So these differences in the conditions led to selection pressures. So there was competition for food, and other new selection pressures arose. Since there was some genetic variation that occurred among the ancestors, so I said genetic variation within the population resulted into advantageous alleles. It means among the population of the ancestors, some of them had some alleles that gave them an advantage to be able to survive in this environment where there are selection pressures, as well as competition for food and maybe so many things. So they had an advantageous alleles. Then I went on to say that some birds survived and reproduced. Those that had the advantageous alleles, they survived, they reproduced, and they passed on the advantageous alleles onto their offsprings. Now, this was repeated for over generations, leading to new species. So over generations, this repeated survival and reproduction led to a generation of new species, which we have today. So the species they have, Amakihi, that has a longer beak, so they are able to drink nectar from flowers, tree sap, spiders, as well as insects because of their sharp, long beak. They also have larger populations because they adapted to live in more habitats. You saw they live in 3 different areas. On the other hand, Palila has a more powerful beak. that can crack open the of course, coats of seeds. That is why they can be able to feed on seeds as well as berries. So if you had written this a little bit less, you would be able to get the marks we're looking for. So I'll just move on to the next part. Here they say, explain how scientists could determine which of the 17 other species of honeycreeper on these islands is the most closely related to the Hawaii pallilar honeycreeper species. What they will do here is to carry out molecular phylogeny. This could be analysis of DNA, analysis of proteins, or RNA, and anything, any biological molecule present if it could be analyzed. After that, they could find out which species have the most similarities among these analyzed molecules, or which species have the least differences. If they have the most similarities or the least differences, it means they are the most closely related. So this brings us to the end of question 5. Let's move on to question 6. Question 6. Fertilization in mammals such as the African northern white rhino results in the fusion of gamete nuclei. The photograph shows the last 2 remaining northern white rhinos. Both of these rhinos are females, so we can see they're female. And then they go on to say, sperm and tissue samples were taken from the last few male northern white rhino before they died. The sperm samples were frozen, and the photograph shows an organelle taken from a sperm cell as seen using an electron microscope. The diagram shows a sperm cell. So this is the organelle, and that is the sperm cell we're looking at. So they asked, in which part of the sperm cell would this organelle be found? This is a mitochondria. It should come from the middle piece, this part here. So the answer here should be a C, because structure Y, that is where it should come from. So moving on, they say sperm cells contain a digestive enzyme called acrosin. The effect of acrosin activity of samples of sperm cell on the percentage of egg cells fertilized was investigated. The graph shows the results of this investigation. So here we have the vertical axis, percentage of egg cells fertilized, horizontal acrosin activity of the sperm cell. Then they say, describe the role of one organelle involved in the production of this enzyme. Now this is the description. So an enzyme is a protein. So any organelle involved in protein production could be maybe a ribosome, could be a Golgi apparatus, could be mitochondria. This provides the energy. It could be a nucleus or rough endoplasmic reticulum. So any of these, you would get the marks you're looking for. However, we had to say the role of each. So for the ribosome, it is the site for protein synthesis. For the mitochondria, it produces ATP required for protein synthesis. And for the Golgi apparatus, it modifies and packages proteins. The nucleus, this is the site for transcription of the genes that are going to be used to make the protein, which is the enzyme. And then lastly, here we have the rough endoplasmic reticulum. This is a site for the formation of the tertiary structure, the 3D structure of the protein, and the packaging of the proteins into vesicles. So any one of these organelles and any one of these roles corresponding to the organelle you have written above would earn you the marks you're looking for. Let's move on. So here they say, the investigation used 2,500 egg cells for each sample of sperm cells. Calculate the number of egg cells that would have been fertilized by sperm cells with 2.5 AU acrosin activity. If you go back to 2.5 from the graph, let's go back. 2.5 is here, and it gives us a percentage of egg cells that were fertilized to be about 32%. So I'll take you back here. If this is corresponding to 32% being fertilized, it means since if we had this at the beginning, 32% of that should be 800 eggs. So 32 divided by 100 times 2,500 gives us 800. The answer should be that. Here they're going to say, explain why a higher activity of the digestive enzyme acrosin resulted in a higher percentage of egg cells being fertilized. Now, if you look back again from the graph, let me take you back. Beginning from here to there, overall I see beginning from 2.5 to about 5, this one here to 5, that is a doubling. From 2.5 to 5 is a doubling. When you double the acrosin activity, we can see this increases from 32 to 100, which is about by 68%. So here I said, overall doubling the acrosin activity from 2.5 to 5.0 increases the percentage of the fertilized egg cells by 68%. This was from 32 to 100. And I said this is because the higher acrosin activity causes faster digestion through the outer layer of the zona pellucida of of most egg cells. Remember, we have so many egg cells, and if there is a lot of acrosin activity, it means so many enzymes are being released and they are working actively, digesting through the layers and enabling the sperms to go through in order to reach the surface of the egg cell. So I say this allows the sperms to bind to the egg cell membrane and enter, and the sperm nuclei can therefore fuse with the egg nucleus. Or with the egg nuclei, if you talked about sperm nucleus to egg nucleus 1-to-1, so that's it. If the acrosin activity is lower, meaning there is less digestion around the egg cells, some sperm cells could die before they can be able to fertilize, or they could be destroyed before they can enter for fertilization to occur. So a lower acrosin activity would lead to a lower rate of fertilization, or fertilization of fewer eggs in comparison to when the acrosin activity is really high. So let's continue. Here they say in vitro fertilization, IVF, has been used to fertilize northern white rhino egg cells using the frozen sperm sample. The zona pellucida of some egg cells were damaged when they were removed from the ovaries of the females. The diagram shows a fertilized egg cell with 3 nuclei. So here we can see 3 nuclei. A fertilized egg cell should have 1 nucleus inside because the sperm cell nucleus and the egg cell nucleus have already fused. If we have 3 nuclei inside, it means there is a problem. Maybe the zona pellucida was not hardened enough, or the region around the cell was not hardened enough to prevent polyspermy. So it means polyspermy has taken place. So let's continue here. They say, state how to calculate the actual size of the egg cell. To calculate this actual size, they've given us the magnification. So what you would do is you would say, we divide the image size by magnification. Remember, they've told you how. So we are looking for how. I also said, remember, magnification is image size divided by actual size. So if you want to find the actual size, you will say this is equal to that divided by that. So just this written in blue could be enough to earn you the 1 mark. Down here they say, suggest why this fertilized egg cell has 3 nuclei. I said it has 3 nuclei because polyspermy has occurred. There are 2 extra sperm nuclei inside that. When a sperm cell enters the egg cell, cortical reactions occur resulting into the hardening of the zona pellucida, and this prevents polyspermy. However, polyspermy has occurred because the zona pellucida has been damaged, or the damaged area around the zona pellucida did not harden successfully. If I take you back here, they said the zona pellucida of some egg cells were damaged. If they were damaged, it means when the cortical reactions occur, the zona pellucida did not harden well, and failure to harden properly could lead to some sperms going through and penetrating the egg cell, leading to polyspermy to occur. So this is the end of question 6. Let's continue to question 7. Question 7. Sweet pea plants were originally found in Sicily and southern Italy. They are used to investigate genetic inheritance and linkage. The photograph shows a flowering sweet pea plant. So here we have that photograph here showing the sweet pea plant. And down here they say, sweet pea plant flowers can be different colors and have a variety of pollen grain shapes. A says, how many of the following statements about a pollen grain are correct? So they contain a tube nucleus and a generative nucleus. This is correct. The generative nucleus divides by mitosis to form 2 female gametes. This is wrong. It forms 2 haploid male gametes. And then it can produce diploid gametes. This is also wrong. The gametes should be haploid. Remember, we have a pollen tube nucleus and the generative nucleus. This divides to form 2 haploid male gametes. So the answer here should be a B. Only one statement is correct. Moving on to the next part, it says, the table gives information about the surface area and volume of round and long pollen grains. So here we have the round as well as the long. They've given us the surface area, volume, and surface area to volume ratio. The first part here they say, calculate the difference in surface area between round and long pollen grains and give your answer in standard form with appropriate units. Remember, we are looking for this, which is surface area, given the surface area to volume ratio as well as volume. So surface area should be volume times the surface area to volume ratio, which gives us that. So it should be 4.128 micrometers squared. However, since these values here were given, I took this part here to be 5.03. So I wrote the answer here in the same. I rounded it off to to three significant figures to call it 4.13 as well. So here they want us to find the difference. The difference should be that minus that, which we get as this. And they want the answer to be in standard form. So 0.902 is the same as 9.02 times 10 to the power negative 1 in standard form. Next, they say, plants that grow from a sweet pea seed contain stem cells. Describe how a stem cell in a plant can become a sclerenchyma cell. So one thing we have to know is for stem cells to become specialized or to change, to be able to form, like in this case, sclerenchyma cells, a lot of modification has to go on. So I said in the stem cell, gene expression takes place, and this can be by histone modification, DNA methylation, and so on. There are so many modification processes that occur, and these can lead to the gene to become activated. So if they're active genes, these are used to synthesize enzymes, and these enzymes or proteins can be able to permanently modify a cell. If that cell is permanently modified, it is going to be able to produce lignin, produce cellulose, produce microfibers, cause secondary thickening, and therefore become a sclerenchyma cell, because a sclerenchyma cell should be able to do all this. To be able to do this, it's because some modification has occurred on its DNA, meaning some genes that are responsible for the production of lignin, for the production of cellulose, formation of cellulose, microfibers, and so on, are going to be activated. Some of the genes that are not necessary will be switched off, but these are going to be activated, and therefore this cell becomes specialized in order to be able to produce these things and become a sclerenchyma cell. So I will continue. Here they say, sweet pea plants were used in an investigation into the inheritance of flower color, pollen grain length, and pea pod shape. Each of these traits is controlled by a single gene. Sweet peas have 7 pairs of chromosomes. The arrangement of these 3 genes on sweet pea chromosomes is shown in the diagram. So we have chromosome number 1 has 2 specific genes they're giving us. There is a gene for flower color, and there is a gene for pollen grain length. On this side, we have a gene for pea pod shape on chromosome 4. So they're going to ask, they say, comment on the role of meiosis in the inheritance of 3 of these traits. One thing we need to know is meiosis causes random assortment as well, or during meiosis, there is random assortment and there is crossing over. So I said, through crossing over and random assortment, meiosis increases genetic variation. And then these genes for flower color and pollen grain length are found in close proximity on the same chromosome. Let me take you back. This is the same chromosome, and they are in close proximity. It means during inheritance or during passing on these genes, these 2 genes are going to be inherited. They will remain in the same or on the same chromosome. They will be inherited on the same chromosome. They cannot be crossed over. So that is why they are on the same chromosome and in close proximity. So here I said, the genes for flower color and pollen grain length are found in close proximity on the same chromosome. We say they are linked. Their alleles are therefore more likely to be inherited together since they are not separated during crossing over. Remember I said crossing over as well as random assortment are going to occur. Now if you compare this one here and those ones, of course, due to random assortment, they can be inherited differently as well. So I say since the gene for pea pod shape and that for flower color or grain length are not linked, not on the same chromosome, they are inherited independently due to random assortment. So this brings me to the end of question 7. Let's continue to question 8. Question 8. Elk are one of the largest species of deer. The photograph shows 3 elk grazing in Yellowstone National Park. They prefer to feed on young tree shoots, such as aspen and cottonwoods, rather than grass. So continuing down here, wolves are introduced into Yellowstone National Park in 1995. So they were introduced during that year. In that year, wolves are predators of elk. Before the introduction of wolves, elk grazed undistributed. So here it means they grazed all over, not in specific areas, but all over the whole area, eating all the young tree shoots in the forest and riverside habitats. The table shows how the percentage of young shoots eaten by the elk changed after the introduction of wolves. So let's see here, we can see as the years went by, we can see the percentage of young tree shoots eaten by the elk In the forest habitat, we can see it decreased, but not as much as what happened around the riverside. We can see there is a greater decrease around the riverside. Of course, maybe the wolves were also there because it's a riverside. There is water, so there are many predators around this area. So the decrease was not as great, was bigger in comparison to the decrease that occurred here. The forest habitat, we expect there were fewer predators in comparison to the riverside habitat, so there is a greater decrease in the percentage of young tree shoots eaten by the elks in the riverside area than in the forest area. So that is what I see overall. Let's see here. They said, explain why the percentage of young tree shoots eaten by the elk changed after the introduction of wolves. So here I said, of course, when they say explain, it means you have to write which fact has occurred or the difference that has occurred, and then give a reason why. So I had to give my statement. I said there was a decrease in the percentage of tree shoots eaten after the reintroduction of wolves. When the wolves were introduced, there was a decrease in the percentage. Then I had to give a reason. I said that elks could not graze freely in the presence of the wolves. Remember, wolves are predators of elks. So more young tree shoots were eaten by the elks in the forest than in the riverside habitat. Also, the mature trees could provide camouflage to hide them. This I mean to hide the elks from the wolves in order for them to survive. So we can see there was an increase in the number of, I'll take you back here to show you properly. Here we can see there is more. We can see here there is a decrease, at least in the number of young trees that were eaten because there are fewer elks around this area. While in this area we can see the decrease was not as great because they had more of this area. So we can see they stayed eating more of these trees in comparison to this one. Remember, this is a percentage of young trees eaten by elks. Less were eaten by elks in comparison to this area here. It's because we had the predators around the riverside area. So let's go back to the next part. Here they say, the photograph shows a beaver. We can see this beaver here. Then they say, the graph shows the mean beaver lifespan in areas with different numbers of elk per kilometer squared. Here we see the mean beaver lifespan. We can see it decreases as the number of elks increase per kilometer. This could be because the beavers and the elks are competing for the same thing. That is why they're decreasing as the elks increase. So let's go on to see what questions they asked. They said, describe the relationship between the graph. This is just to describe. You do not have to give a reason why. So just write what you see on the graph here. On the graph, you can see that overall there is a decrease as the number of elks increase per kilometer squared. There is a decrease in the mean beaver lifespan. That is overall. However, there is a greater decrease around this area. And then a very slow decrease around this area. So those are the 3 points we can see here. And exactly that is what I wrote. I said increasing the number of elks per square kilometer leads to a decrease in the average beaver lifespan. There is a rapid decrease in the beaver average lifespan from 30 elks per kilometer. So from that end, there was a rapid decrease. After 80 elk per kilometer squared, there is a very little change in the average lifespan of the beavers. So there you get your 3 marks. Let's move on down here. This scientist investigated the effect of introduction of wolves on the biodiversity of Yellowstone National Park. Suggests what information the scientists would need to collect in order to calculate species richness in a habitat of Yellowstone National Park. So to be able to calculate species richness, we need to know the number of species that are available, and we need to know the area of the habitat. So get the total area of the habitat and get the number of species that are available in that area. Down here they say, write a formula that could be used to determine the biodiversity of a habitat in Yellowstone National Park. The formula should be this. D, which is the diversity index, should equal to N. Capital N is the total number of organisms in all species into that same capital N minus 1 with the brackets divided by the sum of N. Small n is the total number of organisms in each individual species. And this is summation. You add up everything. So you multiply n into n minus 1 and then add everything to get the total of that. So this formula here should be used to find the biodiversity of a specific habitat. Let's move on. So here they say, Beavers and elk eat the shoots of young trees. Beavers use older trees to build dams in the rivers, which create new habitats such as ponds. Beavers live in colonies of up to 8 individuals. After the introduction of wolves, scientists recorded the number of wolves, elk, beaver colonies for 15 years. They also recorded the number of young cottonwood shoots that were taller than 5 centimeters. The graph shows the data collected by scientists. So here we can see the number of wolves on the vertical axis per year. We can see there is an increase and then a decrease and an increase and a decrease over time. But overall, we can see there is an overall increase from 20 to about 40 from 1995 to 2010. So there is an overall increase of about 20 wolves. Then going down here, the number of elks, we can see overall the elks decreased from 1995 to 2010. Although there are those certain increases, but overall there was a decrease. Then the number of cottonwood shoots over time, we can see from 2003, there is an sporadic or greater increase. As you can see, it's kind of exponential increase from that time. This could be due to maybe the decrease in the number of elks that were feeding on those cottonwood shoots. So if they decrease in number, it means now they have time to be able to grow because there is less predators or less organisms eating them. Then here we have the number of beaver colonies. We can see again from about 1999, the beaver colonies begin to increase, as we can see that. Discuss the effect of introduction of wolves on the biodiversity of Yellowstone National Park. Use all the information in the question to support your answer. So here I looked at all the graphs that were given and the information in the questions. So I observed that overall the wolves increased by 20 wolves from 1995 to 2010. If I take you back here slowly to the number of wolves, you can see overall here we had 20, and at the end of 2010 we had about 40. So that is an increase from 20 to 40 is about by 20 wolves. So that is why I say that. Then also overall the number of elks decreased from 1995 to 2010 because they were hunted by the wolves. Remember, the wolves are the predators and the elks are the prey. Also, the number of cottonwood shoots rapidly increased after 2003. You'll observe that. Again, taking you back here. You can see from here, this is 2003. We can see a sporadic increase in the number of cottonwood shoots, as we can see there. So they increased rapidly. After 2003, this is because the number of elks decreased and thus the cottonwood shoots were not eaten as much. Remember, the elks feed onto these cottonwood shoots. So if the elks are decreasing in number, it means the cottonwood shoots should also be able to increase in number. Next, I say the number of beaver colonies increased from 1999 to 2009. There was an increase from 1 to 12. And the beaver colonies started to increase when the elk population decreased. That is also observed from the data. Next, so this shows that previously the elks outcompeted the beavers for shoots. Remember, they feed on the same kind of thing, or they have the same niche. So meaning they can outcompete each other. One can outcompete the other and be able to survive or increase in number in comparison to the other one. So here I said the decrease in the number of wolves is due to the decrease in the food source. That is the decrease because there were so many wolves. They fed on the elks decreased in number, so the wolves will also decrease in number because they do not have enough food to feed on. Lastly, when the wolves were introduced, biodiversity increased. Initially, there were fewer species. Introduction of wolves introduced a new species, which increased the biodiversity of this area. So this brings us to the end of this video and the end of this paper. Thank you for being with us. Please do not forget to subscribe to our channel. I'll see you in the next video. Bye-bye.